And good morning, everybody. It's a great pleasure to be back at BioKorea again, although unfortunate that we are not able to attend in person due to the pandemic. Many of you have already experienced Cynata Therapeutics, but just for those of you who are familiar with the company, I'm pleased to present the following information. We are developing a proprietary platform for the manufacture of therapeutic mesenchymal stem cells, or MSCs. We call our platform Cymerus, and it involves the use of iPSCs, induced pluripotent stem cells, as the starting material for the production of the therapeutic MSC product. We are the most advanced company worldwide in the development of iPSC-directed cell therapies, and now have multiple clinical trials underway. As we observe, many other companies are now embracing the potential of iPSC strategies for the manufacture of cell therapy products, including, for example, CAR-NK cells and dopaminergic neurons, just as two examples. The company is listed on the Australian Stock Exchange, and our market capitalisation is around $65 million. We are well financed with around $25 million in cash, and we are well supported by a number of major shareholders, including bioscience managers, a major healthcare investment fund, and Fidelity International, an institutional investor with global reach. A strategic partner for one of our products is Fujifilm, and they're just under a 6% shareholding. In summary, Cynata is very well positioned in the regenerative medicine space with a robust and scalable technology for the manufacture of therapeutic MSCs. We have completed a successful clinical trial with very, very good results, which I'll talk more about in a moment. And that has allowed us to expand our activities to a broad and very comprehensive clinical development pipeline. With the excitement around MSC-based therapies, the company has significant growth potential underpinned by its strong intellectual property platform. As I think many of you will be aware, MSCs are probably the most attractive cell therapy being investigated to date. MSCs present broad potential treatment opportunities for a wide range of diseases, but obviously the growing success of MSCs will create demand for a large quantity of product. And ultimately, companies that have a viable, scalable manufacturing solution will be those that are successful. And Cynata is extremely well placed. When we consider the approach to manufacture of therapeutic MSCs, we must look at the conventional solutions that are used today for the production of these products. And for the most part, conventional methods require donor-derived strategies, donors who are prepared to donate bone marrow or adipose tissue. The MSCs are then purified. But as we know now from the literature, MSCs exhibit considerable phenotypic changes during culture-based expansion, and this limits the ability to use these products commercially when we rely on this donor-derived strategy. And in fact, it's very notable to observe that these phenotypic changes and changes in gene expression profile occur in as few as 13 population doublings, which, based on a bone marrow source, provides only about 1.6 doses of MSC. So as you can understand, there is a real constraint there in the ultimate industrialization of MSCs that is holding back, I think, the commercial potential. So, of course, many companies have explored the notion of multiple donors, but this is a very challenging prospect as well. First of all, it's not a practical solution. We just can't have an industrial process based around many donors providing their tissue source for purification and isolation of MSCs. So really the solution then relies on a completely different starting material that allows us to manufacture large quantities of MSCs without relying on multiple donors or without excessively expanding the MSCs themselves in culture. And to illustrate that graphically, this slide shows the conventional process that relies on deriving MSCs from multiple donors, isolating the cells from each donation, expanding the cells in culture, and then using those expanded cells in therapeutic applications. This is the conventional process that nearly every company today in the MSC space utilises. But as I indicated before, that is very challenging. In our case, we utilise a completely different approach that relies upon induced pluripotent stem cells, or iPSCs, as the starting material. iPSCs, as many of you will be aware, are capable of essentially limitless self-renewal. They can be cultured indefinitely and expanded indefinitely. And this allows us to generate a very large quantity of starting material that is then differentiated through our patented process into the finished product. In other words, a mesenchymal stem cell or an MSC. So this ensures, of course, a highly consistent product because we are always using the same starting material compared with multiple different donors, all of whom have different characteristics in the conventional process. So using the same starting material always ensures consistency, and because the expansion, the generation of large quantities of cells for commercial application, is at the level of the iPSC, ensures that we have very fresh and robust, in other words, high quality MSCs as the finished product. And indeed, this has been demonstrated in comparison studies where we've looked at the potency of our cells compared with cells derived from bone marrow or conventional sources. And this issue has become more and more important in the ultimate commercialisation of MSC-based therapies, especially last year with the FDA's commentary around a biologic license application that was submitted to the FDA by one of the manufacturers of MSC products. And that BLA was rejected by the FDA in part as a consequence of issues relating to donor-to-donor variability and the impact that that has upon the heterogeneity of batch to batch and the lack of consistency. So this is an excerpt from the FDA's briefing document, which draws attention to this, as they say, functional heterogeneity, or in other words, a lack of consistency between MSC batches derived from different donors. And fundamentally, this issue will not go away while humans are different from one another, because therefore their MSCs are different from one another. The Cynata strategy addresses this very issue in providing a highly consistent starting material. In developing our technology, it's been important to demonstrate that cells derived using our process are effective. And we recently completed a 2-year follow-up of patients in a clinical trial in graft versus host disease. This has now been published in Nature Medicine. In fact, it was the front cover of Nature Medicine magazine in November last year, and I would encourage you to view this article. But in brief, our clinical trial showed an outstanding result at both day 100 and at 2 years, where we saw a complete response in these very, very sick patients of over 53% and a survival rate of greater than 87% at 100 days, and at 60 days, at 2 years, we saw 60% survival, which compares very well with standard of care. These patients have a very poor prognosis, and less than 20% of patients typically are still alive after 2 years. Even with other MSC products, the survival rate is still quite low compared with our 60%. So this was a landmark study for the company as well as for iPSC-derived cell therapies. This has allowed Cynata to move forward now with multiple clinical trials in graft versus host disease through our licensed partner Fujifilm, in osteoarthritis, in ARDS and COVID, and further trials that we anticipate undertaking in the future, particularly in diabetic foot ulcers, idiopathic pulmonary fibrosis, and renal transplantation. Therefore, Cynata has a very rich and diverse pipeline around its MSC technology. Briefly, the osteoarthritis trial is a phase 3 clinical trial that is currently underway here in Australia in 440 patients. This is a very large commercial opportunity because there is no current treatment for osteoarthritis. It's a highly prevalent condition. Indeed, in the United States, there are more than 30 million patients with osteoarthritis and a global market worth more than $11 billion US. So if this trial is successful, and it's certainly well designed, then this will be a transforming event for not only Cynata, but cell therapies more broadly. We also have undertaken and have underway a clinical trial which we call Amended, which initially was in COVID-19 patients with respiratory failure, but we've now expanded that trial to include patients with respiratory failure from any cause. So, for example, influenza or other respiratory infections. And this is now underway in Australia at several hospitals here. Together, these targets represent significant commercial opportunities for the company. I mentioned already osteoarthritis is a very large target with a high level of unmet medical need, and so too are some of the other targets that we're exploring as well. So finally, to conclude, I'd like to summarise that Cynata is very, very well placed in the growing regenerative medicine sector with a robust and viable manufacturing solution for mesenchymal stem cells that addresses the current challenges associated with a donor-derived strategy. Our technology is scalable and enables the consistent and high-quality production of MSC-based medicines. We have demonstrated the utility of our technology not only in preclinical studies in multiple different targets, but also in our clinical trial in graft versus host disease that had compelling safety and efficacy data. This has allowed us to move forward to a broad and attractive clinical pipeline with multiple clinical trials currently underway and further trials in the planning phase at the moment, allowing the company to have a significant growth potential with attractive commercial opportunities and with the cash currently on our balance sheet in order to effectively execute on our strategy. I'd like to draw your attention to the fact that we are a publicly traded company, so please do refer to the Australian Stock Exchange website and announcements platform, and also our website for further information about the company. And I thank you very much for your kind attention, Kamsahamnida, today, and I look forward to interacting with many of you during the course of the next couple of months. Please do get in touch with me, visit our website and contact me if you have any questions whatsoever arising from our presentation today. Thank you very much.